So these are some of the studies we have done between 2023 and 2025. And we did more. Actually, we developed also multiple sequence aligner, biometrics, and came up with an AGI pipeline for drug discovery and all that. But I will not be taking this, since this is a training workshop, to an advanced level. We will be talking about things that are suitable for the training workshop. So, Jokela type spinal muscular atrophy, bio-inspired polymers for robotics, that means also humanoid robotics. AI-based structure prediction tools are the topics today. And why doesn't it work? So when it comes to Jokela type spinal muscular atrophy, it's a late onset SMA. It's a neuromuscular disease. It includes muscle twitches and cramps and reduced motor function like walking. The genetic mutation of CHD10, which is a protein with intrinsic to the cell regions, is at the center of spinal muscular atrophy. And Manu Jokela discovered that in 2011. So Professor Dr. David Kang and Alexa Jungabu are my collaborators. They do the experimental studies and at Case Western Reserve University, and I do the computational studies regarding this disease. So coiled helix 10 mitochondrial, in short, CHD10, is a protein that has intrinsic to the cell regions and has 149 amino acids. The structure was unknown until we did the first studies, and experiments faced challenges as we talked. X-ray is in solid state, and MR, well, it's an intrinsic to the cell protein, so it's kind of very difficult. It's enriched at crystal junctions in the intermembrane space of mitochondria and interacts with various proteins and organizes the mitochondrial space actually and function. So when it comes to CHD10, we did bioinformatics studies, homology modeling studies, multiple run molecular dynamics simulations, deep learning and thermodynamic calculations. And we published these papers in 2022 and 2023. Here you see the deep learning model that we used. And the structural assembles of CHD10 and the G66V, whereby the 66 residue is mutated, causes the spinal muscular atrophy, Jokela type disease. Here you see the first row is the wild type, the second row is the G66 mutant type CHD10 protein conformations. And these simulations were conducted in water. So here are the snapshots. And the average root mean square fluctuations were calculated, RMSF values. And we saw that the mutation impacts especially the N-terminal region flexibility, but also there were discrepancies in the mid-domain and C-terminal regions. We calculated also the average secondary structure properties. And especially when you look at beta sheet, you see discrepancies between the wild type and the G66V mutant type. And it looked like that the G66V mutant type has the same alpha helical characteristics and similar turn characteristics. But the turn the point structure and the beta sheet varied, and the G10 helix varied as well. So then we calculated based on the radius of gyration, average radius of gyration, which measures the compactness of a protein, right? And the end-to-end distance, the average free energy surface areas. And you see that the mutation which causes the disease has an impact on the free energy surface of the wild type. So even though the radius of gyration and compactness doesn't change very much, the end-to-end distances vary a lot. Then we calculated the Ramachandran plots and the tertiary structure properties. Here you can compare them to each other. And you see small differences in the intermolecular interactions within the CHCHD10 protein between the wild type and the G66V mutant type, actually. Then we calculated the PCA, principal component analysis was conducted, and we saw that the dynamics of these two, the wild type and the G66V mutant, were really different from each other. So when we look at the PCA plots, the cause that we found was because CHCHD10 or the S59L mutation causes the ALS disease, and the G66V mutation causes the SMA-J disease. So when these mutations impact the wild type structure, which in turn impacts the dynamics of these proteins, can these two diseases be related to the protein dynamics? Can they be associated with the protein dynamics differences, was the question. And we found that it depends on the dynamics. Regarding bioinformatics, we calculated the disorder propensity, and we found that CHCHD10, G66V, and the impact of G66V mutation on CHCHD10 were enormous. These are intrinsically disordered proteins. So in summary, I already told our results. But unlike the S59L genetic mutation, the G66V mutation does not impair the mucus complex, and no experiment has shown that G66V mutation alters CHCHD10 aggregation in mitochondria. This is in accord with our computational studies. So our computational studies agree very well with the experimental studies, and this is the power of molecular dynamic simulations in accord with deep learning studies. So another topic that I want to talk about is, this was between the years 2021 and 2025, actually. Nature-inspired engineering of self-healing polymer-based materials for soft-robotic applications. But you can use these polymers, actually, Stojmer, in aquaculture as well. For example, to sample the pollutants, small particles can be used, actually. These are very flexible and they highly coordinate to ligands. So it's a very interesting area. Maybe we should look into that. So natural processes. Nature is replete with examples of processes that through evolution have been perfected over the ages. Efficient energy, matter, space usage, and to sustain life. It's resilient to time-tested environmental changes, able to recover and rebuild when disturbed or damaged. It's transcends barriers of scalability while maintaining efficiency. Its robust, dynamic self-organization and self-healing abilities exist, and human-made formations strive for, yet often fail to achieve these natural processes. So we draw the inspiration from natural processes to create innovative solutions to engineering challenges, actually. Addressing some of the grand challenges in functional materials, smart systems, and robotics via bio-inspired design. In looking at biology, there is an approach for dealing with damaging conditions. Developing soft robotic components out of synthetic materials possessing a biomimetic healing ability would permit the healing of both microscopic and macroscopic damages. In biology, forms and functions are intimately intertwined. Properly designed structural mimics will often be able to perform the functions that you predict. So, the intellectual merit of this project was to develop materials for soft robotics applications via smart polymers that are bio-inspired from intrinsically dissolved proteins. So intrinsically dissolved proteins, as we said in the previous lecture, are highly dynamic. They do not adopt stable structures, and they aggregate. So they come together, and they form oligomers, fibrils, and aggregates. That causes, for example, Alzheimer's disease or Parkinson's disease in neurodegenerative diseases. However, these can be also useful when you look at the bright side. So when you bio-inspired synthesis of these intrinsically dissolved polymers, synthetic polymers, based on the structural properties of intrinsically dissolved proteins, could give us self-healing abilities in polymers, right? And this could open new avenues with new class of smart polymers. But the literature back then had focused on synthesizing de novo tailor-made polymers to perform specific functions. Bio-inspired polymers, protein folding mechanism complexity has limited reliable engineering of specific structures. But intrinsically dissolved proteins have high flexibility and self-organization characteristics. And quasi-foldamers are polymeric molecules up to 50 MERs based on a non-natural backbone and are able to mimic the secondary structure of proteins. So these smart polymers, we thought, can transcend nature by imparting improvement in biostability and bioavailability, and parameters can be controlled for intermediating dissolved structure activity relationship. In a broader impact, minimizing both the financial burden and avoid tissue injuries or even more serious problems typically encountered in the defense industry. It can be used as building blocks in construction industry and natural industry and health industry and aviation industry or in environmental science. So foldamers, what are foldamers? Foldamers have been found to mimic helix turn in sheet-like secondary structures. Examples include alpha-beta peptides, gamma peptides, and substituted glycines, phenylalanine, ethylenes, and urea derivatives. And an alpha helix coil foldamer was reported too. All these non-natural oligomers exhibit kinetic and thermodynamic stability and can mimic peptide functions. Our studies are about mimicking intrinsically dissolved proteins which adopt a function based on ligand binding and self-assembly. So this study, for example, requires an interdisciplinary approach. And we were planning to rationally design and conduct the computational studies of intrinsically dissolved polymers, which we did with Nisa Tan. Thank you, Nisa. The synthesis of new class of bio-inspired intrinsically dissolved polymers, the design of a robotic arm preferred by Bilgem, and characterization of new class of bio-inspired intrinsically dissolved polymers, machining of bulk polymer structure into filaments for 3D printing purposes, and then we wanted to conduct a mechanical characterization of 3D printed polymers. So the structure-breaking amino acid residues are histidine, lysine, proline, arginine, glutamic acid, glutamine, and glycine. Glycine has just hydrogen as a functional group. And these are the synthesis ways that I planned, that I designed. With a new chain extender for the new class of polymers using intrinsically dissolved proteins as models. And in summary, these class of polymers, and Nisa conducted the computer simulations using molecular dynamics simulations, and she found that their properties are very close to intrinsic proteins, actually, of synthetic polymers that are biomixed by intrinsic polymers. So, new class of self-healing and soft bio-inspired intrinsic polymers with multifunctionality can be provided. New soft robotic operating systems can be delivered using these new class of bio-inspired intrinsic called urethane polymers. In addition to the robotic muscles and exoskeletal applications, these new class of polymers may be useful in prosthetic, edible robots, picking, packaging, and in search and rescue applications. So these are our collaborators. And now I will be talking about structure prediction by AI-based tools. Some of the AI-based tools that you are familiar with might be AlphaFold 2, AlphaFold 3, iTester, CI-Tester, ESNFold, RosettaFold, right? And AlphaFold 2 and RosettaFold, well, they got the Nobel Prize. These can be linked to replicate exchange molecular dynamics simulations and NMR experiments. So to study the intrinsic proteins. What is AlphaFold 2? In short, it's developed by DeepMind and Google. It won the CAS14 competition in 2020. A neural network known as transformer is applied. It utilizes multiple sequence alignment, which are collections of similar protein sequences from different organisms, to understand the evolutionary relationship and conservation of amino acid positions. And exactly this multiple sequence alignment in AlphaFold 2, how many of you heard that AlphaFold 2 creates hallucinations? It's hallucinating. 1, 2. Have you heard it? Yes. So few people heard it. How many of you have used AlphaFold 2? Just a few. Okay, tomorrow we will be using AlphaFold 3, which is a newer version. But AlphaFold 2, it uses the multiple sequence alignment, and it uses a conventional one. And after reading it, I thought this was the reason for hallucinations, and we started developing a new multiple sequence aligner. And our results show clearly that it gets rid of, when you use it with AlphaFold 2 together, it gets rid of the hallucinations that AlphaFold 2 causes, and gives more accurate structures for intrinsic disorder proteins. But you have to be very careful in choosing the sets, the sequence sets, or MSA, multiple sequence alignment. So AlphaFold 3, again, developed by DeepMind Google. How many of you have used AlphaFold 3? It's actually only my students. And you. Yeah. So it was introduced in May 2024. It has substantially updated diffusion-based architecture, which is capable of joint structure prediction of complexes, including proteins, nucleic acids, and small molecules, ions, and modified residues. Higher accuracy was reported for structure prediction and protein-protein interactions. And AlphaFold 3 reduces the amount of multiple sequence alignment processing by replacing AlphaFold 2's evoformer with the pairformer module, but it still depends on multiple sequence alignment. So, and then we have iTester, developed by the Divine Group, CI-Tester, which is the contact assisted iterative trading assembly. And then we have Fire 2, and then we have RosettaFold. How many of you heard RosettaFold? It's a deep learning-based algorithm for predicting protein structures, similar in concept to AlphaFold 2. It represents a new approach to the problem, utilizing the principles of deep learning to predict how the amino acid sequence of a protein falls into its three-dimensional structure. Rosetta Fold uses a three-track neural network which processes information about sequences, pairings between amino acid residues, and the three-dimensional topology of the polypeptide chain. So the experimental accuracy was compared to Rosetta Fold, and it was found that it gives accurate results. And then how many of you have heard ESM4 by Meta? Facebook. It's another large language model-based application for predicting protein structures. Okay. So how many of you have heard replicate-change molecular dynamics? Wait. So this is a parallel tempering molecular dynamics simulation technique. The problem with molecular dynamics simulations is, I will explain that after lunch, but it can be checked in the minimum energy area of the surface when you look at the energy surface, like the conformational energy surface, right? It can be checked in the minimum and cannot sample all conformations when it has a rough surface, for example. So replicate-change molecular dynamics simulations are a special sampling technique. It's different than classical molecular dynamics simulations. And you have different replicas, multiple replicas or copies of the system that are simulated in parallel in this technique. And you play around with the temperatures. We will talk about that more, but it enhances the conformational sampling of the system. So when it comes to how good these AI-based models are regarding intrinsically disordered proteins, I found that all of them failed. And I would like to thank Lisa Tan for her studies, and Jahid and Melissa. Didn't have your pictures, so I couldn't put it here, sorry. And so the MSA technique that we developed, which is called biomimetics, is especially important since it reduces the hallucinations of these AI tools when they are used together, like AlphaFold 2. And we hope to come out with more algorithms, actually, on which we are working currently. And this was the presentation. So you can go to lunch. It's 12 o'clock. After lunch, we will start with molecular dynamics simulation.